Assalamu alaikum. Today we are going to learn about the Kreb cycle. It is called a Kreb cycle because it was discovered by a scientist named Kreb. It is also called a citric acid cycle because the first product formed in this cycle is a citrate. It is also called a tricarboxylic acid cycle as well because the product which is formed in the Krebs cycle citrate is 3 carboxylic group. That's why it is called a tricarboxylic acid cycle as well. It provides an exceptional insight for the cellular energy production. It takes place in the mitochondria. It produces ATP energy when the NADH and FADH produced in the Krebs cycle provide electron to the electron transport chain. It acetyl-CoA which are produced as a result of pyruvate oxidation, and it produces carbon dioxide and water as a byproduct, and it generates high energy electron, as I previously said, NADH and FADH2, and it is essential for the cellular energy because it provides these electron rich species to the inner mitochondrial matrix to get going the electron transport chain. Now we will move on to the cycle. Here we got the acetyl CoA from the pyruvate oxidation, and citrate synthase is an enzyme which acts in the acetyl CoA and oxaloacetate, which is previously produced in the previous Krebs cycle, and it will produce the citrate. Here you can see we got two substrate, acetyl-CoA and oxaloacetate, a 2 carbon and a 4 carbon molecule. Citrate synthase will act in these two substrates, acetyl-CoA and oxaloacetate, and will produce the citrate, which is a 6 carbon molecule. Now we have another enzyme, aconitase. Aconitase enzyme will act on the substrate citrate, and it will remove water from the citrate, and it will produce the cis-aconitate. But here is isomerization take place in the third step, and the aconitase enzyme will act on the cis-aconitate, and it will add a water molecule, and it will produce the isocitrate molecule. The isocitrate will act as a substrate for the isocitrate dehydrogenase enzyme. This dehydrogenase enzyme will remove hydrogen from this isocitrate molecule, and it will produce the alpha ketoglutarate, and that hydrogen which is removed by the dehydrogenase will reduce the NAD molecule to NADH, and one carbon dioxide molecule is removed in the process. Now the alpha ketoglutarate dehydrogenase enzyme will act in the alpha ketoglutarate and remove hydrogen, and that hydrogen will reduce the NAD plus to NADH, and one carbon dioxide molecule will reduce in this step, and at the end we got a product called succinyl coenzyme present in the mitochondrial matrix will add a CoA to the succinyl substrate. Now the succinyl CoA synthetase enzyme will use and utilize this succinyl CoA substrate and will remove the CoA from this succinyl and produce the succinate. Here one inorganic phosphate will phosphorylate GDP into a GTP, a guanosine triphosphate. Now the succinate dehydrogen enzymes will utilize the succinate substrate and remove two hydrogen atoms, and those two hydrogen atoms will reduce FAD into FADH2. As a result of this reaction, we got a fumarate. Fumarate is a substrate for an enzyme fumarase. Fumarase will add the water molecule to the fumarate and produce amylate. Now we got amylate. Amylate dehydrogenase enzymes will utilize the malate and produce oxaloacetate. As a result, hydrogen is removed from the malate, and that hydrogen will reduce one another NAD plus to NADH. At the end of the Krebs cycle, we got oxaloacetate. What will happen to this oxaloacetate? It will wait for another acetyl CoA which must came from the another pyruvate oxidation, and when they got acetyl CoA, oxaloacetate and acetate synthase, it will fuel and it will start another Krebs cycle, and it will go on, and at the end we will got another oxaloacetate molecule, and it will move on again and again. Let's move on to the energy yield or financial step of the Krebs cycle. We got a 3 NADH, 1 FADH2 and 1 GTP molecule at the end of the Krebs cycle. But you can see here in the illustration that we got 6 NADH, 2 FADH2 and 2 ATP molecule per glucose. So let's revive the glycolysis cycles that we start the glycolysis cycle with just one glucose molecule, and we got at the end a 2 pyruvate, a 2 3 carbon molecule at the end of the glycolysis cycle. So if we got 2 pyruvate molecule at the end of the glycolysis cycle, one pyruvate molecule will be oxidized to the 1 acetyl CoA. So if we got 1 acetyl CoA, it will fuel just one Krebs cycle. But we got another pyruvate molecule from the glycolysis cycle, that another pyruvate molecule will again oxidize into another acetyl CoA molecule, and it will fuel another Krebs cycle. Therefore, if you got two pyruvate molecule from the glycolysis from one glucose molecule, then you will surely get two acetyl CoA molecule. Then two acetyl CoA molecule will fuel and start two Krebs cycle rather than one Krebs cycle. That is why we can say that we got 6 NADH and 2 FADH2 and 2 GTP molecule instead of 3 NADH, 2 FADH2 and 1 GTP, because this process is repeated, and this process is repeated due to the 2 acetyl CoA molecule availability. This is all about the Krebs cycle, the citric acid cycle, or we can say tricarboxylic acid cycle. Uh, this is just short illustration and short summary of this Krebs cycle. You can get a critical hit and blow at each and every step of this cycle, because this is a complex process and this is a very extensive process. You will get a lot of thrill, exciting and surprising critical question when you are studying this process extensively, not for just only exam. You want to study it, you want to master it, this process, you will get a lot of question. That is why I would recommend you to master it, to learn it, to study it, not for only exam, but for the thrilling and exciting feature you got after this whole process. Do ask some critical question in the comment box. It will be my pleasure to discuss those questions, to answer those questions of you. Thank you for watching and listening this exciting features and events of the Krebs cycle.